My name is Ray Corrigan. I'm a PhD candidate in biomedical engineering. I work in the Michael Schnieders lab within ITI at the University of Iowa. And the work that I do focuses on solvation. So every molecule in your body is in some sort of environment, and I believe all of them are in a solvated environment. So to be able to represent that computationally is essential to being able to simulate or computationally model the function of those molecules within our computer models. So the work that I do is trying to improve computational solvation models. So these are representations of the forces of water on various different biomolecules to make sure that those forces accurately represent the forces and the interactions of water that these molecules will be experiencing in your body. This has broad-reaching applications. I'm working on nucleic acids and proteins. We've done some work with small molecules also, so tiny things like methane or imidazole, and hopefully we'll be able to continue to expand the sphere of applications there.
I'm Rose Goebel. I am a second-year PhD student in Dr. Michael Schnieders' lab, and I study the biochemistry of proteins, specifically with titratable amino acids. So amino acids, certain ones can have different protonation states, so there can be a hydrogen or there cannot be a hydrogen, depending on the environment of that residue. And so what I do is I try to use a global optimization algorithm to predict whether that hydrogen exists based on the protein's environment or that specific residue's environment that can include the other residues within the protein.
Hi, I'm Mallory Tollefson, and I work for Professor Michael Schnieders in ITI at the University of Iowa. I'm currently a doctoral candidate, and I'm actually looking at deafness and the causes of deafness. So deafness is a heterogeneous disease. It's caused by many different genes and many different mutations within each one of those genes. So oftentimes when a patient has deafness, they have up to 500, maybe even 600 mutations that could be causing their deafness. So what I'm doing right now is I'm developing protein structural models where I can model different mutations that patients have in order to look at how a protein changes according to those mutations and try to figure out which mutations are causing deafness.
I'm Michael Schnieders. I'm a joint faculty member in the Department of Biomedical Engineering and the Department of Biochemistry. My lab works on understanding the genetics of a number of diseases, including hearing loss, renal disease, vision loss. And our approach is to make atomic resolution models computationally, and we focus on everything from the underlying physics and statistical mechanics to writing the software, running the simulations, and then interpreting the simulations to understand why is it that a genetic change could lead to a disease.